So I'm Omar Sabari, currently finishing my PhD at the Technion. And today I'm going to talk about scalable and robust DNA-based storage via coding theory and deep learning. This is our recent work that was published in Nature Machine Intelligence. And this is joint work with Daniela Bar-Lev, Itai Or, Tuvi Etzion, and Eitan Yacobi. So I'll start with the model or the pipeline that we were trying to solve, and this is I would say the most standard in vitro DNA data storage pipeline. I'm sure you're all familiar with. I'll just briefly go over it to make sure that everybody is aligned. So we have some binary data which is encoded into short DNA oligos. Then these DNA oligos are synthesized using some synthesis technology. And was kind of the right procedure of our storage system. And when we want to read back our data, then we take a sample from the synthesized oligos, we perform PCR and sequencing, and then we get many noisy copies of the original encoded sequences, of the original sequences that encodes our data. And the problem that we solved in this paper was to find a retrieval algorithm, including an encoder and decoder, that can handle these many noisy copies and retrieve back the data from them. So the model we assume is a base level substitution, deletion, and insertion errors. Again, in deletion, for example, this T was deleted from the sequence. In insertion, we have an insertion of T here. And of course, most of the sequences or of the sequencing grids can get more than a single error at the same time. So common approach to tackle this problem or to actually correct these kind of errors was to use inner and outer code. So in inner code, we encode each designed strand separately using some error correcting code that correct errors at the nucleotide level. And in outer code, we encode all the strands collectively to overcome missing strands or some strands with like many errors, very noisy strands. So again, this is a very simple and very flexible method to correct errors in DNA data storage. However, it's also not very optimal in terms of redundancy, and it requires high coverage, meaning that we need to write many reads in order to get enough reads to encode the sequences. How many? So I calculated here according to our scheme. Let's say that we want to store 1 terabyte of digital information in DNA. We're gonna need 30 billion DNA strands, each of length 200 bases. And we're gonna need like a half trillion sequencing grids in order to be able to correct these errors. And of course, also in terms of pricing, the pricing are becoming more attractive, but still very pricey compared to other storage medium. So this is something that we need to consider when we design our system, and this is kind of what we did. So our approach was to first cluster or bin the noisy copies into clusters, and then take each cluster of noisy copies and perform reconstruction on it, kind of try to correct most of the errors by using the inherent redundancy that we have from synthesis and sequencing. And then we have to correct the remaining error using our decoder, meaning that we can now use much less redundancy in our decoder or in our encoding scheme. So I mentioned clustering. So clustering is a known word from machine learning and deep learning, very known task in machine and learning and deep learning. But in DNA, this task is a bit different, mostly because the number of clusters is very high. As I mentioned, it can reach to billions of clusters that we need to form. And also the distance metric, in our case, at a distance, it's not differentiable, and it's time consuming to compute it. So for clustering, we kind of bypass the clustering problem by presenting a very simple and efficient binning algorithm that simply bin each read based on a predefined index that we encoded in the encoding phase. And of course, we use indices with certain distance from each other to make sure that even in case of errors, we can still bin the reads correctly. Still, we had some more noise in clustering because we use like very simple algorithm, but this is something that we were able to overcome in the reconstruction procedure. So I mentioned reconstruction. So reconstruction is actually a class of problems in which there are several noisy copies of the information. And the goal is actually to decode the information. This problem was also studied in many theoretical aspects, kind of asking what is the minimum required number of clusters. But in our case, the number of copies is given, and we should design an algorithm in order to correct or to use the inherent redundancy to correct the errors in the copies. So for reconstruction, we use transformer-based deep neural network. The input is actually all the noisy copies of the clusters, and we kind of put them into a one-hot embedding, into a matrix that represent each location, what is the different kind of bases that we see in each location. And we use padding in order to make all the sequences to be in the same length. And the output is actually some kind of soft output that tells us in each position what is the probability to have each one of the bases. And we also use this soft output to make kind of a confidence mechanism, which I'll explain later. So the first question was how to train this deep neural network, because as I mentioned earlier, making data in DNA is still relatively pricey. So we kind of overcome this challenge by using simulated data for training. So what we did is that we first ordered a small pilot pool of only 1,000 clusters. We synthesized them, we sequenced them, we performed an error characterization to see what was the deletion, insertion, and substitution rate. And then we were able to generate many simulated clusters that have the same error profile, and we use them for training. So for training, we only use simulated data. And one more interesting attribute of our DNN is that it has two branches, each one kind of taking the cluster from different direction. The first one takes the cluster from the standard direction, and the other one takes the cluster from the reverse direction. This is kind of a mechanism that we added in order to overcome synchronization errors and error propagation that can cause in case of deletion and insertion. So I mentioned a confidence filter. So the confidence filter is another mechanism that we use to make sure that in some cases we can have clusters with much more errors. And these clusters are likely to have an erroneous estimation. So using the confidence filter, we can detect them. We can kind of see that these clusters has low confidence in terms of reconstruction. And for these kinds of clusters, we have another algorithm, which is now based on dynamic programming and not deep learning. And this algorithm actually does not assume anything on the error probability or anything about what's happening there. It just calculates an estimation of what was the error probability based on the reads, on the observed reads, and then it kind of create some kind of weighted graph in each each error event is modeled with specific probability, and then it uses the graph in order to make its own estimation, which can be more accurate for erroneous cluster. And of course, the idea behind balancing these two with a confidence filter mechanism is to kind of balance accuracy versus speed. So let's go to the results. So in terms of reconstruction accuracy, we improved all the previously published algorithms by up to 40% of accuracy. Here we represent the error rate, so lower is actually better. And you can see that we tested our reconstruction method on all of the previously published data sets. In terms of running time, we were able to improve 3 orders of magnitude for the reconstruction. And this is an estimated time for reconstructing 100 megabytes of information. And of course, we also tested our own data set, our reconstruction on our own data set. And again, there we were also showed the highest accuracy. One more important aspect in designing DNA data storage system is to, again, to balance between the code rate or the information rate, the number of bits that we can encode per base. And of course, this is kind of a function, or it depends on what is the channel error probability. So you can see that in our case, the channel error probability, since we use nanopore and we use like the previous hardware of nanopore, was relatively high, roughly 5% error rate that we had to correct. But on the other hand, our information rate was relatively high, especially when considering the channel error probability. And this is one more graph regarding the safety margin mechanism and the confidence filter, but I'll skip it for now because we don't have time. But just in few words, this graph actually shows that when using the confidence filter and when using the reiteration of reconstruction with the CPL algorithm, with the dynamic programming algorithm, we were able to correct more clusters, and by this we were able to retrieve the information even with lower coverage. Okay, so one more aspect of our work is also to design an encoder-decoder that can actually address these specific clusters, and including or considering the fact that most of them will be corrected using the reconstruction algorithms and the clustering and reconstruction algorithms. So our encoded scheme first kind of assess or consider the fact that most of the clusters are correct, and we will have small numbers of clusters with small number of substitutions. And I say substitution because we know that the DNN and the CPL algorithms returns output of the correct length. So therefore, even though that there are edit errors, we can infer them as substitution errors, as Hamming errors. And of course, there are also missing clusters and clusters with large number of substitutions. So what we use is we use a Tensor-Product-based coding scheme in which we kind of have an inner code, but not for all the strands, but only for small fraction of the strands that we can actually select them based on the result. Like during the decoding, we can actually detect which one of the strands has either small or large number of errors. Then we can try to correct them using our inner code of the Tensor-Product code. And then, in case that there are still remaining errors, and also in cases that we have some missing clusters, we can actually correct them using the outer code. And by this, we can actually reduce the redundancy by using Tensor-Product code and by kind of considering the fact that not all the clusters will have or will likely to have errors. So we also published, along with our paper, data, relatively large data of both nanopore and Illumina sequencing. We encoded in our experiments both image, audio, random bits, and text all together into different files. And we also published our pilot datasets. This is the way we design the information. We have primer, we have 12 base for indices, and we have 128 bases for data and redundancy. And this is a barcode in case you want to take to see the datasets. We also published our dataset also in binned format, meaning like post-clustering, and we believe that this can be like helpful in case that you want to benchmark your result or your algorithm and to allow easy comparison between our work and all and all the other work. So now we have like standard bin format for the data sets. And of course, we also published our code in a Git repository in which you can see how we generate the simulated data, an implementation of the DNN, implementation of the CPL algorithm, and again, of course, our encoding and decoding algorithm, and you can scan it also and see the code. So to summarize everything, so we have improved the accuracy by up to 40% of a previously published reconstruction algorithm on previously published datasets and on our own datasets. We have much faster reconstruction algorithm. We use only simulated data to train our model. Again, we were able to correct clustering errors because we use very noisy clustering method, the binning algorithm. And we also combine it with the coding scheme that improve the code rate, the information rate. And thanks for these four wonderful guys that were part of this work, of this amazing work. And thank you.
Thank you. Thank you, Amr. Questions, comments, notes? While everyone is thinking, I have a question for you, Amr. So the simulated data that you used, was it an encoded data or random data?
That's actually a great question. We kind of we try to use both of them, and from some reason it works better when we use like random data. So of course we use it. We use clusters with the correct design length with 128 bases, but we simply generated them randomly according to, of course, the error profiles that we show. So didn't have to encode them before like making the noisy copies and the noisy clusters.
Thank you. Thank you for the answer. Anyone? No questions? Then thank you, Amr. Thank you very much. And we move on. Oh, no, we have a question. Sorry.
Thank you. Thank you for the presentation, Omer. It was very nice. I have a question regarding the data sets. Have you had to retrain your model according to each data set, or was the training done only once?
So that's a great question, and we had to train the model separately per, I would say, sequencing technology, because we use always the same synthesis technology. So per pilot, we had to train different model because there were difference compared to Illumina and nanopore sequencing.
Okay, so for unique sequencing technology, it was not retrained for several times.
Yes, I guess, for example, if we use like the current hardware of nanopore sequencing, which is a bit less noisy, so we need to train it again.
Okay, thank you.
No more questions? Last call? Okay, then this time for sure. Thank you, Amr.
Thank you.